In this module, I will be discussing type 4 hypersensitivity reactions. As a reminder, type 4 is the fourth of 4 hypersensitivity reactions that we have discussed. These are hypersensitivity reactions mediated by T cells. And T cells can mediate hypersensitivity reactions, both T helper cells and cytolytic T cells. T helper cells largely mediate their pathologic effects through the release of inflammatory cytokines. Cytotoxic T cells, of course, can also directly mediate tissue damage by killing cells. One of the classic examples of a type 4 hypersensitivity reaction is the PPD test, or the purified protein derivative test. This is the test used to detect whether somebody has been exposed to Mycobacterium tuberculosis. And it's done by injecting some PPD antigens, some tuberculosis antigens into an individual. And these antigens can induce what's called a delayed type hypersensitivity reaction. And this happens when macrophages take up the antigens and they release inflammatory cytokines that leads to the recruitment of memory Th1 cells that are specific for these Mycobacterium tuberculosis antigens. These macrophages can then present these antigens to these memory Th1 cells that migrate into these tissues, and these T cells will make interferon gamma, which in turn enhances the activation of the macrophage, and this leads to a localized inflammatory response and induration of the skin. So when you get the TB shot, the PPD test, you go back 2 or 3 days later, and they read your reaction and to determine whether you've had a reaction to these PPD antigens. If you have no Th1 memory, of course, then you don't generate this hypersensitivity reaction. And again, this is called a delayed type hypersensitivity, largely because it takes 1 to 2 days to induce the T cell response that results in this reaction. There's other hypersensitivity reactions that also occur in the skin that are somewhat analogous. And these are reactions, for example, to things like poison ivy or a component of poison ivy called urushiol, as well as nickel. And these exposure to these substances can lead to the recruitment of Th1 cells and cytotoxic T lymphocytes that can mediate tissue destruction. It's thought that these compounds react with self-proteins to then induce a compound-specific T cell response. And once again, this reaction requires prior exposure. So you have to be sensitized first. You have to induce a memory T cell response. And then upon the second exposure, those memory T cells can induce the hypersensitivity reaction. And again, this is Th1-mediated inflammation as well as cytotoxic T lymphocyte-mediated killing of keratinocytes, resulting in a blistering rash. And it's often called allergic contact dermatitis, but I want you to be careful. This is not a type 1 hypersensitivity. Contact dermatitis is a type 4 hypersensitivity. And here's just an example of kind of some of the blistering rashes that can occur upon being exposed to some of these substances and getting contact dermatitis. Another classic example of a of a T cell mediated hypersensitivity is rheumatoid arthritis, and this is largely mediated by Th1 cells. It induces chronic inflammation in the small joints of hands and feet. Larger joints can be involved, and this is usually in women 3 to 1 over men over 30, and results in synovial swelling, granulation in the joints, rheumatoid nodules, elevated inflammatory markers, fatigue, and fever. And you'll be learning a lot more about rheumatoid arthritis next year. There are a number of ways to treat it. NSAIDs are common, corticosteroids can be used, and TNF blockers can be used as well. And there are other mechanisms of hypersensitivity that are present. Type 2 and 3 disease mechanisms may also be present. And there are autoantibodies present, including rheumatoid factor and anti-CCP antibodies. And just as a reminder for you, rheumatoid factor is an anti-IgG IgM that's present not only in rheumatoid arthritis, but in a number of autoimmune diseases. This is an example of what it looks like. Here's an IgM molecule that is binding to self-IgG, especially the Fc region of IgG. And so this is an autoreactive antibody. Okay. Now, Th1 cells drive rheumatoid arthritis pathology in part through the production of tumor necrosis factor. And so here we have a T helper 1 cell that is making tumor necrosis factor. It can induce activation of macrophages within the joints that themselves can also then produce tumor necrosis factor. And tumor necrosis factor will have a variety of effects. It can act directly on synovial fibroblasts to induce inflammatory pathology and synovial thickening. It can act on endothelial cells to induce vasodilation and enhance the recruitment of inflammatory immune cells. And it can act on macrophages and T cells to kind of induce a cycle of inflammation within the joint. Now, there are a class of molecules called TNF blockers. Many of these are monoclonal antibodies, and I've listed a few of them here. One of them is called etanercept. This is not a monoclonal antibody, but rather is a fusion protein in which the extracellular portion of the TNF receptor has been fused to the human Fc of IgG. And this basically acts as a decoy receptor to block the action of TNF, and so you block its ability to mediate all these effects within the tissue. There's other strategies to block other cytokines. IL-17 blockers are not useful for rheumatoid arthritis, but they can be very effective in some other diseases, including psoriasis. And so it's thought that diseases that are susceptible to TNF or IL-17 blockers are very likely T helper-driven diseases. There are a number of type 4 hypersensitivities in the gut that induce inflammatory bowel disease and celiac disease. One of those inflammatory bowel diseases is Crohn's disease, and this is largely mediated by Th1 cells and Th2 cells that produce a number of inflammatory cytokines. And this disease really gets going due to a loss of tolerance to gut antigens. As we've learned in the mucosal immunology lecture, there's a balance between regulation and immune activation within the gut that needs to be maintained. And when that gets out of balance, you can get tissue pathology. And so this is dysregulated in the case of Crohn's disease in favor of Th1 and Th17 responses. And this leads to a number of lesions in the large and small bowel and can lead to obstruction. And this is one of those diseases that is linked to the MHC molecule HLA-B27, although it's not really understood how this linkage is contributing to the immunopathology of the response. A second example of inflammatory bowel disease is ulcerative colitis. Again, this is also not an autoimmune response, but again, a loss of tolerance to gut antigens. And this is thought to be mediated, at least in part, by dysregulated Th2 cells, leading to mucosal inflammation in the large bowel and bloody diarrhea. And again, this is also linked to HLA-B27. And then a third hypersensitivity in the gut that I wanted to mention is an autoimmune disease called celiac disease. And so this can be a disease to self-antigens and also to food antigens such as gluten. And so this is a type 4 reaction of Th1 cells that are gluten specific. Also a type 2 reaction may be participating here. There are autoantibodies specific for gluten, as well as one of your self-proteins, transglutaminase. And this leads to foul-smelling diarrhea and villus atrophy in the small bowel. And this disease, like many autoimmune diseases, is tightly linked to MHC class II. Now, besides Th1 and Th2 cells, Th17 cells are also important mediators of autoimmune disease. And I've shown as an example here multiple sclerosis. And so here are Th17 cells that have been induced that are specific for components of the myelin sheath. And microglial cells and macrophages within the CNS can present myelin antigens to these Th17 cells, which can then drive a cycle of inflammation within this site that leads to destruction of this myelin sheath and the resulting symptoms of multiple sclerosis. This is mediated in large part by neutrophils and other immune cells, can lead to death of oligodendrocytes. There's a relapsing remitting form of MS and a chronic progressive form of MS, and it's thought that these different forms are largely due to whether T cells are persistently activated or whether there are waves of activation over periods of time. But it's not really clear during relapsing and remitting MS what triggers those waves. Okay. Now, besides T helper cells, cytotoxic T cells can contribute to many types of type 4 reactions, including type 1 diabetes. And so here we have a islet of Langerhans. This islet is full of insulin-producing beta cells, and these beta cells can be destroyed by insulin-specific cytotoxic T lymphocytes that kill beta cells, as well as Th1 cells that are specific for insulin that can orchestrate inflammation-mediated destruction of beta cells. It's thought that autoantibodies may also play a role in contributing to tissue damage. And again, there's a number of environmental triggers that aren't fully understood for this disease, for type 1 diabetes. But we do know that it's genetically linked most tightly to MHC class II, as well as a number of other genes that regulate immune tolerance. It does have higher incidence in men in this case, but disease tends to be more severe in women. Again, the hallmark symptoms are diabetic ketoacidosis. And one of the treatments for this that's rather interesting is the possibility of getting an islet transplant. There are a number of hypersensitivity reactions that are less well defined, that we don't know for sure what type of hypersensitivity is driving immune pathology. An example of this is ankylosing spondylitis. This is an inflammatory arthritis of the sacroiliac joint that leads to systemic inflammation. Common in young men. It's another one of those diseases that's linked to HLA-B27, although the reasons for this are totally unclear. TNF and IL-17 blockers are actually effective for this disease, and so it's thought that there's probably a role for CD4 T cells in disease pathogenesis. Reactive arthritis is symptomatically very similar to ankylosing spondylitis, inflammation in the sacroiliac and knee joints. There are infectious triggers for reactive arthritis, and so if you get infected with one of these diseases and develop these symptoms a few weeks later, then reactive arthritis could be one reason. And again, the reason for this linkage between these diseases and the development of arthritis are not clear. And again, this is also linked to HLA-B27. And reactive arthritis is responsive to NSAIDs and TNF blockers as well. And the last example I'll show you is Sjogren's syndrome. This is a secretory gland disorder that can lead to dry mouth, dry eyes, dry skin, dry mucosal surfaces. Autoantibodies are present, but it's not clear what role they play in pathogenesis. There's also lymphocytic infiltration of the secretory glands, but again, not clear what role they play in pathogenesis. So this is an autoimmune disease, hypersensitivity reaction, but the mechanisms of disease are not yet fully understood. Okay, so take a moment to read over the summary here to make sure you're gleaning what you need to out of this short lecture.